Hey everyone, welcome. Today we are diving deep into one of the most important techniques in modern biology. It's a method that literally lets scientists listen in on the secret conversations happening inside our cells. And it all starts with a really simple but kind of profound question. So think about this for a second. Every single cell in your body, from a neuron in your brain to a cell in your skin, has the exact same genetic blueprint, the same DNA. So how on earth does one cell become a brain cell firing off signals while another becomes a skin cell creating a barrier?
Well, the wild part is the answer isn't in the DNA code itself, but in how that code gets read. It's all about epigenetics. You can think of it like there are millions of tiny protein switches sitting on top of the DNA, and they're constantly flipping genes on and off. They're telling each cell which pages of the instruction manual to read and, just as importantly, which ones to ignore.
Okay, so that brings us to the big challenge for researchers, right? I mean, with 3 billion letters in the human genome, how can you possibly find the exact location of every single one of these protein switches? How do you even begin to create a map of this unbelievably complex system?
And that is where our star player for today comes in, ChiP-Seq. It's a mouthful, I know. Chromatin Immunoprecipitation Sequencing. But what it does is just so cool. It's a powerful method that does exactly what we need. It combines the super-specific targeting of antibodies with the raw power of modern DNA sequencing to create a precise genome-wide map of where all these proteins are binding to our DNA.
So why has ChiP-Seq become such a go-to tool for scientists? What makes it so special? Let's take a look at what makes it so incredibly powerful.
Okay, what's really cool here is how these advantages all build on each other. First, ChiP-Seq gives you incredible specificity. It pinpoints the exact protein you're looking for. But it's not just finding it in one little spot. It gives you genome-wide coverage. On top of that, it's super versatile. You can use it on all kinds of different species and samples. And, maybe most importantly, it's quantitative. That means you can actually measure and compare how protein binding changes, say between a healthy cell and a diseased one.
You know, to really get why that specificity is a game changer, let's compare it to another popular technique called ATAC-Seq. Now, ATAC-Seq is great, don't get me wrong. It answers a broad question. Which parts of the DNA are open and accessible? It gives you the big picture. But ChiP-Seq asks a much more focused question. Exactly where is my specific protein of interest binding? It needs that special antibody to work, but in return, you get that laser-focused answer. So we know why it's powerful. But how does it all, work? Let's walk through the entire journey from a tube of cells all the way to a finished data map.
All right, let's break this process down. First, you take your cells and you add a chemical that basically freezes all the proteins right where they are on the DNA. Then you chop up the DNA into smaller, more manageable pieces. Step 2 is the real magic. You add a specific antibody that acts like a tiny, powerful magnet, and it only grabs onto your one target protein and the little piece of DNA that's stuck to it. After that, you purify just that DNA, get it ready, and send it off for sequencing, where millions of these fragments get read all at once. Finally, computers map all those little pieces back to the genome, and that's how you find the exact spots where your protein was hanging out.
So, after all that work in the lab, what do you actually get? What does the final result even look like, and what does it tell you?
And this is the payoff. The data gets turned into a map with a bunch of peaks. Just think of it like a mountain range. A flat area means your protein wasn't really binding there. But a big, sharp peak, that's a hotspot. That's a place where your protein was binding like crazy in a whole bunch of your cells.
So the big question is, what can you do with this amazing map of peaks? Well, a ton. Researchers use it to map out the entire networks of master regulator proteins to understand how cells develop. They can look for special chemical tags to find hidden control panels in the genome. And, this is a huge one, they use it to figure out the epigenetic changes that drive diseases like cancer, which is absolutely critical for discovering new drugs. It's really a cornerstone of modern biology.
Now, as you can probably guess, a technique this powerful and this precise, well, it's also got its complexities. Getting from your raw sample to a clean, publishable discovery is a major, major challenge.
And this is the question every single researcher has to ask themselves. That workflow we just looked at has a bunch of steps, and at every single one, things can go a little bit sideways. Tiny little variations can create noise and just totally hide the real biological signal. So, what does it actually take to get it right?
Success really boils down to getting these key details perfect. First and foremost, number one with a bullet, is the quality of your antibody.
A bad antibody is the #1 reason these experiments fail, period. Then you need proper controls to tell your real signal apart from the background noise. The DNA fragmentation has to be in that Goldilocks zone, not too big, not too small. You need to sequence deep enough to actually find your peaks reliably. And finally, you absolutely have to run replicates to prove your results are real and not just some random fluke.
So, when you look at all of those variables together, it becomes pretty obvious that this isn't just about following a recipe in a cookbook. You have to understand the nuances at every single step to get data that's plain and robust enough to actually get published. It takes real hands-on expertise.
And that's exactly where having an experienced partner or lab becomes so critical. I mean, you see it right here in this quote from a provider like CD Genomics. They're not just talking about doing the experiment. They're talking about optimized workflows and quality control designed specifically to meet the high standards of scientific journals. That's the level you need to be at.
So what does that expert help actually look like? Well, it means they help you tackle all those challenges we just talked about. They'll help you pick a proven antibody, so you don't waste time and money on a failed run. They use protocols that have been fine-tuned over years of experience. They handle all the complex data analysis to turn those raw reads into real insights. And they guide the whole experiment to make sure your results are reproducible and totally solid.
And that really brings us full circle. With a tool this powerful, a tool that can generate such a precise map of what's happening on our DNA, the possibilities are just immense. From understanding the deep roots of a disease to engineering better crops, ChiP-Seq gives us the blueprint. So it makes you wonder, right? What biological mystery would you want to solve?